Hello, hello. Welcome back to our last lecture on lipid chemistry. Continuation from the lecture from before. We're still talking about the categories of different lipids. And so now we're going to jump into the phospholipids. Those phospholipids, right? Um, let's start out by talking about them. You've heard of phospholipids before, especially if you've taken biology class, right? Because phospholipids are the thing that compose the phospholipid bilayer of cell membranes, right? But let's talk about those chemical structures a little bit here. Get my little script here ready. Here we go. All right. So, phospholipids are in a family of lipids, right? That are similar in structure to the triacylglycerols, right? But they include the glycerol phospholipids and the sphingomyelins, right? So here's your glycerol phospholipids right here, and they have a hydrophobic head group with a phosphate connecting to a glycerol backbone to two fatty acids right down here. Okay? The hydrophilic head group of a glycerol phospholipid has either a choline attached to it, an ethanolamine attached to it, a serine, an isotal or glycerol attached to it, right? So that's where the glycerol phospholipid part comes from, right there, the glycerol, right? And then for the sphingomyelins, right, there's also a hydrophilic head group. The X there, or there, I use the word, I like to use R, but X there is a choline, and it's connected to a phosphate, right? They connected to the sphingosine backbone. That does include a fatty acid chain, right? And then another fatty acid, right? Another fatty acid. Excuse me. I think I just got an email. Let's mute that. There we go. So, just quick review. Glycerol phospholipids. Sorry, I got a little bit distracted by that. But in from my email, glycerol phospholipids contain two fatty acids that form an ester bond with the first and second hydroxyl groups of the glycerol. Right, very similar to the triacylglycerols. Right, because in a triacylglycerol you wouldn't have the phosphate group right here. Right, you would just have another fatty acid coming down, right, from the glycerol. A hydroxyl group that forms the ester with the phosphoric acid, which forms another phosphodiester bond with an amino alcohol, right, which is right here. So, oh, that's a new word right there. A new functional group. New functional group, a phosphodiester bond. Or a phosphodiester, right? That's something new. And what is a phosphodiester? It's when you have a phosphorus, like you see right there, connected to an O, connected to an R, right? Phosphodiester. And usually it's connected on both sides. That's why it's called a diester. Let me just draw that one more time. It's called a diester because it's usually like this. There we go. Right. So in the case of the glycerol phospholipid right there, right? So we'll call that R1 and R2, right? Rest of the molecule 1, rest of the molecule 2, right? And in the case of the glycerol phospholipid, right, this part here of the phosphodiester bond is the R1, and then this part here, which with the hydrophilic head group, is R2. Right? And in between we have our phosphodiester bond with the P of the phosphodiester bond. Right? Now, if you're really good, you'll know where else we see a lot of phosphodiester bonds. And if you can tell me before the end of the lecture I don't know how you're going to tell me. Maybe you can email me or something like that, right? I just thought that that might be very unfair, though, because some people might watch the lecture before other people, and then they won't get a chance at this particular reward. So I'll just go ahead and tell you. Phosphodiester bonds are actually very prevalent in DNA because your DNA is made of a phosphodiester backbone, right? Interlinked with sugars, actually, riboses. But we'll talk a lot about those phosphodiester bonds and those riboses when we get to the DNA part of our class. Then we have our sphingosines that are attached to glycerols instead of, let me say that again. Our fatty acids are attached to sphingosines instead of glycerols and sphingomyelin, right? Like we were talking about here. And it contains one fatty acid, a phosphate, and an amino alcohol, right? Interesting stuff. So what are these hydrophilic head groups that are present in glycerol phospholipids? The ones that I mark as X in the previous slide, right? What are some of those? That's a terrible looking X. Let me try that again. There we go. That was the hydrophilic head groups. Of the glycerol phospholipids, right? What were some of those? Well, there's choline and there's irine, which is not real because that's a typo. That should be serine. That's an amino acid, right? Not irine. And then we have ethanolamine, right? That's some of them. And they are actually considered hydrophilic because at physiological pH of 7.4, they're ionized, which means they're charged, right? So looking at choline here, we have a charged nitrogen. And looking at serine here, we have another charged nitrogen. And looking at ethanolamine, we have another charged nitrogen. And that's why they're considered, or they're called, the hydrophilic head groups, right? Or X in my previous picture. So let's see them in actual glycerol phospholipids, right? One of those glycerol phospholipids is lecithin. Lecithin has a choline head group that's polar, right? It's got its phosphodiester bond right there, connected to its glycerol right there, and the nonpolar fatty acids are there. And this is a 1 plus 14 is 15, plus one more is 16. That's a palmitic acid. Or palmitate, right? Another glycerol phospholipid is cephalin, right? Cephalin means head, so I'm guessing that's in the brain. Its hydrophilic head group, or X, is ethanolamine, connected to its phospholipid. Its phosphodiester bond, and its glycerol. And it also has palmitate as its nonpolar fatty acid tails, right? So let's take a closer look at the structure here of glycerol phospholipids, right? Remember that glycerol phospholipids always have a polar head group. So in that case, it's a choline. So that's lecithin. It has the phosphodiester, but before it joins, it's actually just the phosphoric acid. It has the glycerol, and it has the fatty acid tails. And this is actually a mixed fatty acid because it has a kink right there, right? So we have a saturated fatty acid up here, and we have an unsaturated fatty acid there, right? So largely, though, we consider that phospholipids are composed of two parts. The polar head, polar because the phosphate here is negatively charged, and the choline here is positively charged. And then we have the nonpolar fatty acid tails. Right? Fatty acid tails. That's what that little drawing is there. So it even has little nonpolar tails for you right there. And there's some implications there because since this is nonpolar, the fatty acids are nonpolar, these are what we call, or what we consider, hydrophobic. Right? And since these are polar, they're definitely soluble in water, we consider these hydrophilic. Alright? So hydrophilic, polar head group, soluble in water. Hydrophobic, nonpolar tails, not soluble in water. And so these have interesting implications. I was about to write out the word interesting, but I don't know why you would need me to write that out. Interesting implications when it comes to the cell membranes of your body, your cell membranes, right? And here are those implications. No, I'm just kidding. This is just, it's related, right? So snake venom, turns out, right, is actually composed of phospholipid lipases, right? And if you recall from a previous slide, I was talking about how lipases, or maybe it was a previous lecture, I was talking about how lipases in your digestive system break down the triacylglycerols that you consume, and they break them down into their substituent fatty acids so that they can be transported throughout your body to your adipose tissue for storage, right, in the bloodstream. Well, snake venom has phospholipid lipases. So what do you think those do? They break down the phospholipids that compose the cell membranes of your red blood cells, which is bad because that will destroy your red blood cells. You won't be able to transport oxygen anymore, transport nutrients to the rest of your body anymore. That's a bad thing. So getting bitten by a snake and being poisoned with snake venom is a bad idea. But of course, we have antivenom, which counteract those effects, right? Counteract those effects. But that is how snake venom works. It hydrolyzes the phospholipids in the cell membranes of your red blood cell. So another group of phospholipids are the sphingomyelins. Sphingomyelins. It's hard words to say. Anyway, and you can see in the structure of these sphingomyelins, they're very similar to the glycerol phospholipids, except that you have this thing that is the sphingosine backbone, right? And the sphingosine backbone is this part right here. Right? Oh, sorry, missed this carbon right here. So that's a sphingosine backbone, right? And if you notice, it's a little different from a glycerol phospholipid because in the glycerol phospholipid, this was an oxygen, right? And in addition to having the hydroxyl there, there's also this long chain alkene on the sphingosine backbone, right? So sphingomyelin is composed of the sphingosine backbone, and then the fatty acid, here in this case is palmitic acid again, 16 carbons long. The phosphodiester bond right there. I just spelled phospho with an F. That's terrible. There we go. Phosphodiester. Alright. And then we have the choline. Hydrophilic head group. Right? So these guys are abundant in the white matter of your myelin sheath surrounding the nerve cells. And it increases the speed of nerve impulses, insulates and protects nerve cells. Right? So that's where sphingomyelin is. Then we have our steroids, right? Steroids usually consist of 3 cyclohexane rings and 1 cyclopentane ring fused together. These rings are designated A, B, C, and D, as you can see here. A, B, C, and D, right? And the reason why it's called a steroid nucleus is because all steroids have this part, and then they just have other things coming off of them as substituents and such, right? We always number the carbons beginning with carbons in ring A, right? And then the 2 methyl groups are always at position 18 and 19 for the steroid numbering system. Not much else there to say about steroids, except for the fact that cholesterol is a steroid, right? And you notice here that it has the A, B, C, and D rings, and it has hydroxyl substituents here and this alkane substituent there off the D ring, right? So cholesterol is the most important and abundant steroid in the body, and it has a hydroxyl group at carbon 3. It has a double bond between, oh, I forgot to mention that, it does have a double bond between carbons 5 and 6. The 2 methyl groups are there like they were in the steroid nucleus in the previous slide. And then the alkyl chain, that's connected to carbon 17, right? So, they are present in meat, milk, and eggs. They're synthesized in the liver, and it is needed in cell membranes, brain, and nerve tissue, and steroid hormones, and vitamin D. But it also clogs arteries when there are high levels of plaque form, right? So here we have a nice clean open artery, and here we have an artery clogged by cholesterol plaque. You definitely don't want that. A little more about cholesterol here. It is considered elevated if the plasma cholesterol exceeds 200 milligrams per deciliter, right? It's synthesized in the liver and is obtained by foods. So let's take a look here. Beef liver has, 370 milligrams of cholesterol in every serving size. Good thing I don't like beef liver, right? I do like eggs though. One egg has 200 milligrams of cholesterol. Lobster less. Fried chicken, oh my gosh, I didn't realize fried chicken had less cholesterol than meat and eggs. A hamburger even has less. Chicken with no skin and fish, salmon, they're all very low in cholesterol, right? Skim milk only has 5. Margarine has no cholesterol, but butter has cholesterol in it, right? Because it's the milk and the butter. Margarine's mostly made of oil, if you recall, right? So what are biosalts then? It turns out those are cholesterols too, right? So here's the structure of a biosalt, specifically sodium glycocholate, right? And it consists of a cholic acid here, right, attached to the amino acid glycine here. It's sodium glycocholate because of the sodium that's there. So this is the sodium salt of the amino acid glycine, right? They're synthesized in the liver from cholesterol, and what they do is that they make fat more soluble in water, so it helps with the absorption, right? It helps with the absorption. But the problem though is that if there's too much cholesterol in the gallbladder, gallstones are formed, and I hear that's not very pleasant, right? But so a lot of people have to remove their gallbladders, and when they remove their gallbladders, they're not able to actually process and absorb fat as easily because of the lack of the biosalts that assist in the that act like soaps to assist in making the fats more soluble in water, right? So I had a professor once that used to love having butter dipping sauce with her lobster, and she had her gallbladder removed, and it was not a good time for her when she forgot that she couldn't eat that um, that butter for that lobster. All right, let's talk about lipoproteins in more detail. If you remember what I was talking about when I spoke about it before, lipoproteins are what are your body uses to transport the substituent fatty acids from your triacylglycerols that are broken down by your digestive system, right, through the bloodstream. Well, how does it do it? Well, consider this is the fatty acid that was broken down from the triacylglycerol that you consumed, right? And it was broken down by lipases in your stomach and your small intestine by the lipases into its substituent fatty acids, and it's attached to then a cholesterol, right? So this entire thing is called a cholesterol ester. These cholesterol esters are all combined with each other, right, inside of it's almost like a cell membrane, except it's not a bilayer, it's just a single layer, right, of phospholipids, right? And so what happens is that when all these cholesterol esters are surrounded by a phospholipid layer, right, that also has some cholesterols interlaced in between, it makes them more soluble in water so that they can be transported through the bloodstream, right? And so that's essentially what a lipoprotein is, right? That's essentially what a lipoprotein is. A lipoprotein is basically membrane proteins and phospholipids and cholesterol assembled in a single layer membrane, right, to basically make a ball, capturing inside the ball the broken down triacylglycerol that's been attached to a cholesterol for transport through the bloodstream, right? Because it makes it more soluble in water. Not all of it is broken down triacylglycerols that are broken down into fatty acids. There are also some whole ones too, right? Whole triacylglycerols. But the point of a lipoprotein is to surround a molecule, like a fatty acid molecule, and make it more soluble in water so that it can be transported through membranes and the bloodstream to either cells for usage as energy or to be used to be stored, right? So let's talk about the different types of lipoproteins. There are the chylomicrons, the high density lipoproteins, low density lipoproteins, and the very low density lipoproteins, right? And by density, we mean that by number too, right? We mean that by its actual density. High density lipoprotein on one end of the spectrum is 1.06 to 1.21 grams per milliliter, whereas a chylomicron, or even a very low density lipoprotein, it goes as low as less than 1 gram per milliliter, right? Why is that? Well, let's take a look at its composition by mass. The chylomicrons are mostly composed of unbroken down triacylglycerols, right? And the rest of it is the phospholipids that compose the membrane, the cholesterols in the membrane, the cholesterol esters and the cholesterol esters inside, and the proteins in the membrane, right? So chylomicrons are basically all triacylglycerols that haven't been broken down by your lipases into their substituent fatty acids. Very low density and low density lipoproteins, right? Again, very low density lipoproteins, very high percentage of triacylglycerols, right? Only 8% phospholipids, cholesterol, and protein. Those are the things that compose the membrane, right? And then 12% cholesterol esters. That's the broken down fatty acids from triacylglycerols that have been attached to a cholesterol molecule. Looking at regular low density lipoproteins, this is not mostly triacylglycerols, right? This is what these are what really transport your fatty acids that have been broken down from what you've eaten, right? And that's why it's mostly these cholesterol esters, right? And then 22% membrane, phospholipids, cholesterol membrane, and 22% protein. Now, what makes HDL so good as compared to LDL? Turns out that it's mostly protein, right? 55% of a high density lipoprotein is protein, and then 24% is phospholipids. So the vast majority of this stuff is like membrane, right? And only 15% cholesterol esters, so very low fat, right? Very low cholesterol, and very low triacylglycerols, right? And that's why they say health practitioners and dietitians and nutritionists all say that it's good for you to have a lot of HDL and very little LDL, right? Because LDL is mostly the fats that you eat that come from those triacylglycerols, right? And there's very little of that in HDL, right? High density because, and you might be asking yourself, what? I don't understand what this is here, because it says high density and there's not as much fat. Well, actually, it's high density because it's more protein, right? And protein weighs more than fat, and that's why it's higher density than low density lipoprotein, which has less protein and more fat, right? That's why people always say, when you're working out and you're building muscle, you never lose any weight, and that's because muscle weighs more than fat does. Well, the same is true in lipoproteins as well, right? So shoot for those high density lipoproteins; they're good for you. So, let's say you're eating a piece of bacon. That kind of looks like bacon. And it goes down in your stomach. And the bacon fat, in the form of triacylglycerols, are broken down into their substituent fatty acids. And those fatty acids are attached to cholesterols. And those cholesterols are grounded up into LDLs, right? And they go straight to your heart and start clogging up your coronary arteries. Right? Bad idea. Bad idea. Get some more HDLs in there, right? Get some more HDLs in there. But generally, though, lipoproteins such as HDLs and LDLs transport nonpolar lipids and cholesterol to cells in the liver for either energy storage, right, or fat storage, or usage, right? So let's quickly go through some hormones, some steroid hormones. Sorry, let me say that again. Let's quickly go through some steroids, right? The first steroids we're going to go through are the hormones, the steroid hormones. We have testosterone here, estrogen here, progesterone here, and norethindrone here. Excuse me, right? And we all know that testosterone, progesterone, and estrogen, right, are sex hormones. And then we can see here that they look just like steroids because they have the A, B, C, and D rings. It's interesting though because estrogen has a aromatic ring in the A ring, right? But all these other ones have double bonds in the A ring, just single double bonds in the A ring, right? And other substituents making them different, right? So then we have the anabolic steroids, different from the hormone steroids, right? Or the steroid hormones. And these are actually all derivatives of testosterone, and they're illegally used to increase muscle mass, but they have bad side effects like fluid retention, weird hair growth, sleep disturbance, and liver damage, right? But again, these are all just steroids, A, B, C, and D, right? They all have it. And these are particularly difficult to say. Methodienone, right? You see here back to our organic chemistry naming, own, right? Because it has a ketone right there, right? Then we have oxandrolone, which has an own there because it has a ketone right there, right? Then we have nandrolone and stanozolol, right? Still a steroid though. It has another 5-member ring over here, but it's still a steroid because the steroid center is still there. Steroid nucleus, right? A, B, C, and D. Next, we have the adrenocorticosteroids. These help regulate different systems in your body. For example, we have aldosterone, which regulates the electrolytes and keeps water balance by the kidneys, right? And then we have cortisone, or also known as glucocorticoid, right? And that actually increases blood glucose level, and stimulates the synthesis of glycogen in the liver. And if you don't remember what glycogen is the glucose storage molecule in your body, right? It turns glucose into glycogen when there's insulin. So here's cortisone, also steroid, A, B, C, D, right? Here's aldosterone, helping with those electrolytes. Cortisol, and one that you should all know, prednisone. How many times have you had to take prednisone because of allergies or something, like a bad allergic reaction or something? I mean, I've had to take a cortisone shot every once in a while because of my bad allergies. But these are the things that we use, right? Because they're actually immunosuppressants. Steroids, corticosteroids like cortisone and prednisone are immunosuppressants. And when you have allergies, that's actually an overreaction of the immune system, and that's why they give you corticosteroids, because they suppress the immune system. Next. Only a couple slides left, and we're going to now finally talk about the cell membrane. As you all probably know from biology class, right? Freshman biology class, our cell membranes consist of a phospholipid bilayer, right? So you can see here in this picture here, there's the hydrophilic head that's water soluble, that likes water, and then the hydrophobic tail, right? If you recall from a previous slide, right, this is what we usually see, and this is the hydrophilic head. Right, like a choline or something, or an ethylamine. And then these are the lipid tails that are hydrophobic. Hydrophobic. There we go. What does that mean? Well, these are nonpolar on this end, and these are polar on this end. Which means that it likes to be around water on this side, and it hates to be around water on this side, right? So naturally then, these phospholipids will arrange themselves so that the polar side, right, is facing towards the water, right, facing towards the water. But this is different from the lipoproteins, because the lipoprotein only had the single layer of the phospholipid, and then on the nonpolar side was those fatty acids, right, with the cholesterols, right? And so that was pretty nonpolar, and so it was fine being on the same side as the hydrophobic nonpolar tails, fatty acid tails. But for this to be a bilayer, the other side is another phospholipid flipped upside down, so that its nonpolar side, right, is over here. Sorry, its polar side is over here. Except that you can't see because my head is in the way, so let me change the view real quick. There we go. Its polar head is on this side, right? And now there are two sets of nonpolar tails right here, and they're facing each other. Why is that good? Well, that's good because now you can have water on both sides, right? So inside the cell membrane is your cytoplasm over here, and your nucleus, and your all the different organelles that are essential for cell function. And then over here is the outside of the cell, where it's also water, right? And it can receive different nutrients and proteins and hormones and such like that, right? So that's what it means by a phospholipid bilayer. So what's really interesting about that, though, is that there's this thing called the fluid mosaic model of the cell membrane, and what it actually means is that everything that's positioned inside the cell membrane is very fluid. It's not static. It moves around throughout the membrane, right? And you can't see me because my picture is gone, but I'm moving my hands around very fluidly. Let me just switch back so you can see. There we go. Very fluidly. Everything in the cell membranes are moving around very fluidly, right? And that includes things like cholesterol, right? Receptor sites for hormones and transport proteins, right? And they're positioned in those cell membranes in such a way so that the parts of them that are afraid of water or hydrophobic, right, are anchored to the center of the cell membrane where it's also hydrophobic, and the parts that are hydrophilic are exposed to the water sides of the phospholipid membranes, right? So that it makes it easy for the cell to control what enters and leaves the cell through these proteins, right? Through these transport proteins. All right? So the thing you got to remember about the cell membrane, right, is that it is a phospholipid bilayer, right, where the polar heads of the phospholipids are exposed to the water side on one side of the cell membrane. Then on the other side of the head, right, are the lipid tails, fatty acid tails, and they butt up against more fatty acid tails and another polar head of another phospholipid, right? And just imagine all these just surrounding around a little bubble of water to make a cell. And then outside the bubble of water is also of the membrane is also water, right? Where there's nutrients floating around and such, right? And so you can get nutrients from outside the cell, inside the cell, by having transport proteins or signal things, instruct the cell to do things using receptor proteins, right? Receptors and things like that, right? And then the organelles inside the cell are safe in the nice little hydrophilic area of the cell, right? That's all enclosed by the phospholipid bilayer, right? So it's much more complicated than that because there's an entire class that I took called cytology, and it's all about the cell chemistry and such. But you just have to remember how the cell membrane is composed, right? Once again, phospholipids heads are exposed to the water sides that are hydrophilic. Tails are facing each other, right? That are hydrophobic. And then there are proteins and receptors and carbohydrates and all kinds of other things that are floating around very fluidly in that membrane, right? For cell function. So it contains proteins, carbohydrates, and cholesterol, which I already said that. Has unsaturated fatty acids that make the cell membrane fluid rather than rigid, right? And has proteins and carbohydrates on the surface that communicate with hormones and neurotransmitters. Yep. But that's all I really want you to know about the phospholipid tail. How is this different from the other one? Remember, I spoke about this just a second ago. these are very similar to high density, low density lipoproteins, except the high density lipoproteins and low density lipoproteins only single layer, right? So it can't be water on both sides. It's water on the outside and then hydrophobic on the inside, and that's where the fats stay and they get transported through your bloodstream that way, right? Just making sure you remember that. And then our very last slide, a little bit more detail about transport through cell membranes, right? There's diffusion, which is also known as passive transport. Particles move from a higher concentration to a lower concentration, and we've talked about this before with osmotic pressure, right? Being hypotonic, isotonic, or hypertonic to the surroundings, right? There's facilitated transport, which uses protein channels to increase the rate of diffusion. So that's how the cell gets different materials from outside the cell inside the cell that are specifically needed, and that's with the transport proteins. And then there's active transport, which moves ions against the concentration gradient, right? So just real quickly here, with this picture, we have here diffusion, which is passive transport. And when the concentration inside the cell, right, is less than the concentration outside the cell, then diffusion will happen so that the concentration inside the cell becomes more like the concentration outside the cell, right? Because right now, it's hypertonic outside the cell. And it wants to become isotonic, which is same on both sides. Now, if we're opposite and there was more on the inside of the cell, then it would go the other way, right? Because then you would be hypotonic outside the cell. Right? And then we have our facilitated transport right here. And that's done by a protein that has a special little channel, right, that goes between the two layers of the phospholipid bilayer, right? And it transports specific molecules into the cell. And then there's active transport, where it requires energy, right, to actually push things like waste outside the cells, right? And that is very cursory how transport through cell membranes work, right? All right, that ends the lecture. Just again, we have Blackboard assignments 12, and quizzes 10 and 11 due by the end of the day today. And then by the end of the day next time, on Tuesday, April 13th, Blackboard assignments 13 to 14 are due, and quiz 12 is due. You guys have a wonderful week, and I will see you next time.